Hello, everybody, and welcome to today's Revive webinar from the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I am hosting this webinar on test tube to patient PK/PD of fixed-dose beta-lactam, beta-lactamase inhibitor combinations. For those of you who are joining our webinars for the first time, Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website. I encourage you all to visit the Revive website to stay up to date about future webinars and watch recordings of previous ones. Right now, you can already register for our 4 next webinars. As always, today's presentation will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel, as shown on this slide. We will address the questions after the presentation, and we'll do our best to respond to as many as possible. Today's speaker is Vincent Tam from the University of Houston College of Pharmacy in Texas, USA. The webinar will be moderated by Shampa from the University of Liverpool. Welcome, Shampa. I now hand over to you for a brief introduction.
Thank you. I'm delighted to introduce Professor Vincent Tam. Vincent Tam received his Bachelor of Science degree from the National University of Singapore, followed by his PharmD from Albany College of Pharmacy in New York. After completing an infectious diseases pharmacy residency and also a clinical pharmacology fellowship, he is now a full tenured professor at the University of Houston College of Pharmacy in Texas. He's a board certified in pharmacotherapy with added qualifications in infectious diseases. Vincent has authored and co-authored over 160 peer-reviewed publications in antimicrobial pharmacokinetics and pharmacodynamics, as well as infectious disease therapeutics. He is on the editorial boards of Antimicrobial Agents and Chemotherapy and Journal of Global Antimicrobial Resistance. He has also served as a grant proposal reviewer for the NIH and an ad hoc reviewer for more than 20 journals, which include the International Journal of Infectious Diseases, Journal of Antimicrobial Chemotherapy, and Lancet Infectious Diseases. I'm very privileged to hand over now to Vincent for his presentation.
Thank you, Shampa, for the kind introduction. Fellow colleagues, ladies and gentlemen, good afternoon, or good morning, or good evening, wherever you are. I understand that this is being shared with many countries across many continents. So depending where you are, welcome. I'm glad that you're able to join us this afternoon. Let me see if I can advance my slide. Okay, so what I'm going to do is, before I start, I want to thank the organizers for the opportunities to share some of our work with you. And I have no disclosure to related to the material that I'm going to present today. For the next 40 minutes or so, I hope to achieve the following objectives. Hopefully, you will have a better recognition on the value of beta-lactam, beta-lactamase inhibitors. I'm just going to use BLI for the rest of my presentation. This combination, you recognize the value of these combinations. Hopefully you will have a better understanding on the limitations on the way that how we do susceptibility testing currently. I will share with you some of our ongoing work in the laboratory now, and hopefully you would have to appreciate the potential utility of a novel approach as to how we can go forward and do things faster, more accurately, and more efficiently. So this is what I aim to do today. But just for the students and the trainees on the audience, I just want to make sure that everybody is on the same page. So this is my only background slide there. Combination therapy is very commonly used in clinical practice, especially for the treatment of tuberculosis and HIV infection. It will be considered as more or less the standard of care. Now, on the other hand, for the treatment of bacterial infections, this is less well accepted, relatively speaking, and for fungal infection as well. But due to the increasing prevalence of emergence of resistance, increasingly we are left with no choice. The clinicians are really backed up, and we don't have a choice, and we are increasingly using combination therapy as our last viable resort, especially against some of those multi-drug resistant or pan-drug resistant gram-negative bacteria that I have listed on the slide. All right, so now when we're talking about a beta-lactam, beta-lactamase inhibitor combination, there are essentially two components of therapy. The first part is really on the beta-lactam, and we have had lots of experience. We have used these agents for decades, and the PK/PD of these agents are relatively better understood because we have a lot of experience using these agents. Most people, most experts in the field would recognize that these agents are what we would call time-dependent agents, in the sense that the rate of killing is saturated relatively easily with about 4 to 6 times MIC, and it's really the time above which the threshold during a dosing interval that is really closely linked to its bacterial antibacterial activity, as you can see here. So if you look at a dose fractionation design, you can see that comparing the time above MIC, the correlation to the effect is much more closely characterized as compared to whether you are using AUC to MIC or the peak to MIC ratios. So that's the easy part. Nobody is really struggling developing or using the beta-lactams clinically. The tricky part is when we add on a second agent, in this case a beta-lactamase inhibitor, this is where we start to struggle. Now, for the most part, I know there are exceptions out there, but for the purpose of the discussion now, we are going to assume that most of these inhibitors lack intrinsic antimicrobial activity. They don't really work on their own. I know the exceptions, and we can come to that maybe towards the end of the presentation, how we handle those exceptions. But for the most part, they don't really have intrinsic activity. Their purpose is really when used in combination with a beta-lactam, the purpose is really to potentiate or really to restore the activity of a partnering beta-lactam agent. So they don't have activity, but there is to bring out or to restore the activity of the partner. And in a way that they would reduce the MIC or restore the MIC to a susceptible range based on concentration or time, just like the beta-lactam can kill based on a time dependency or a concentration dependency in a very similar framework. What they do is they reduce beta-lactam MIC, and depending on the potency, depending on the PK/PD characteristics, they can do so based on concentration or they can do so by time. The real problem, when the rubber meets the road, is that traditional PK/PD concepts are not really directly applicable in this setting. And specifically, there is no such a thing as a fixed MIC. I will spend a lot more time later on towards the end of the presentation highlighting this fact. So this is not a straightforward relationship. When I first came across this topic, I thought it has turned out to be much more difficult and complicated than when I first started. And I will spend more time explaining my struggles for the past decade later on. Right, so I have a table listing some of the common products that we have used clinically, that are being used clinically in the US. You can see that there are several proprietary products on the market, but there are two striking features. Number one is that the beta-lactam agent is really locked in with a particular beta-lactamase inhibitor. The BLI is not freely available for clinician to mix and match, so to speak. They are locked in the fixed combination, agent combination. And we will spend some time talking about that later on. Is it a good way or is that a better way, a more flexible way to go forward? So first, the agents, the BL, the beta-lactam, and the beta-lactamase inhibitor are locked in a fixed combination. At the same time, although there are different several pharmaceutical formulations available, the dose of the beta-lactam to the dose of the beta-lactamase inhibitor are also locked in a particular fixed ratio. Now, if you look across different products, there is no one fixed ratio. You can see that for piperacillin tazobactam, there are 3 formulations available, but they are all in the ratio of 8:1 in terms of the dose of the piperacillin to the dose of tazobactam. You can see for the rest, there are several products on the market, but there is no one fixed ratio. So depending on the manufacturer, depending on the sponsor, different programs will have come up with a different number as to what is optimal. Okay, keep that in mind. We'll come back to this later on towards the end of the presentation. We'll shed some light on whether there is a good thing or this is a more flexible way of doing things. All right, so I'm going to first start off with some of the lessons that we have learned a long time ago, just to give you a perspective of where we started decades ago. So when we're looking at piperacillin tazobactam, arguably the most commonly used BL-BLI combination, at least in the US for sure, that what we have learned during the drug development was that the clinical trial was not very successful and not very favorable outcomes was observed, and the FDA, as part of the regulatory process, they mandated a warning, a statement that has to be inserted into the package insert that a higher dose of piperacillin tazobactam must be used, and you should use it in combination with an aminoglycoside, because especially for Pseudomonas, not very favorable outcomes were observed during the clinical trial. But you can argue that that was a long time ago, and we have made a lot of progress over the past several decades on PK/PD. Fair enough. I'll give you another more recent, more contemporary example. So we are looking at ceftazidime avibactam. This was a breakthrough drug, and this drug was available, contingently approved by the FDA before phase 3 trial was even started. So this was a special arrangement because of a lot of issues going on and a lot of people were dying. So there was a contingency plan that was implemented, allowing the drug to be available clinically while the company undertook the phase 3 trial. But surprisingly, shortly after the drug was released, we already found that resistance to ceftazidime avibactam is already emerging. It was very surprising to many of us. We thought this was going to be the agent that would save millions of lives, but we did not expect resistance to be developed so quickly. And also, colleagues have also identified pneumonia and renal replacement as risk factors for failure for these drugs. So from the PK/PD perspective, resistance, pneumonia, and renal replacement, to me, these are all signs that would suggest that perhaps the dosing has not been vigorously examined, because these are all typical pharmacokinetic issues that we consider whether we have the right dosing exposures where we need to do the job. So there are signs of concerns, and I'm just going to take the very conservative position saying that perhaps after decades of having these agents and with the recent advances in PK/PD, we are still not doing a perfect job. There's still room for improvement on how we can do better in terms of understanding the PK/PD and how to develop these drugs in the clinical pipeline and how to use these drugs clinically. All right, so many of you know that I have multiple roles to play during my daily activity. As a clinician, sometimes I work with clinicians, students to see patients, and I work closely with some clinicians in the hospital as well. And I also work with microbiologists, and of course, I hang out with a lot of the PK/PD scientists, the PK/PD geeks. for different issues. And over time, I have learned many things from these colleagues, and quickly I realized that although we are all looking at the same problem, depending on the background of these colleagues, they may have a very different perspective of how they see the problem. So I'm going to start off with the clinicians. They are very stressed and they're under a lot of time pressure because there is a very small window of opportunity, and that pharmacological interventions might actually be meaningful. We don't have weeks and weeks for us to figure out the mechanism of resistance so that we can come up with a rational treatment course. We have a very short time that we need to quickly figure out what is the viable solutions so that meaningful decisions can be made by that side. So time is of essence. I also hang out with many microbiologists, and they have taught me many things over the years and keep my ideas very grounded for practical applications. The number one concern is almost all the microbiologist colleagues are severely understaffed. And whatever that we came up with must not be labor intensive. Simply, there are not enough people and personnel around to dedicated to handle a very laborious procedures specifically for one patient or for one medicine. So whatever that we do pretty much has to be amenable to automation in order to have a chance to survive clinical application. And finally, the PKPD geeks are the one that was making a lot of trouble. You can see that we're asking for the most information on capturing the drug behavior. We want to understand how the bacteria behave and respond to the drugs. We want to understand the influence on infection side, immunology, how the body is fighting back. We want the protein binding issues, blah, 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 blah. All at the end of the day so that we can make better prediction, okay? So very obviously that these three groups of colleagues have very different perspectives, even though we're all looking at the same problem. And very quickly, I realized that it's impossible to make everybody happy, okay? So I gave up on this long time ago. But instead of trying to make people happy, I have turned my research program in slightly differently. I tweak it in such a way that instead of making anyone happy, I'm trying not to make anyone terribly unhappy. So I'm trying basically finding a sweet spot, considering concerns and perspective of different colleagues in order to achieve a common goal. So this mentality, I want to share with you this mentality because this is going to reflect in many of the features that I'm going to show you towards the end of the presentation of some of the ideas that we have. It's basically based on the idea of keeping something that is useful, but also scientifically useful, but also that has a chance to be applied clinically. All right, so when we're talking about combination therapy, I cannot simply ignore the checkerboard method. Okay, so many of you in the audience will probably know this very well. Just bear with me for a minute. So in order to do combination therapy, one of the most common methods is to use the checkerboard method. Essentially, you build a grid with increasing concentration of the drug on one axis. And this could be a beta-lactam or it can be a beta-lactamase inhibitor. It doesn't beta-lactamase inhibitor. It doesn't really matter. And on the other axis, you increase the concentration of another agent on the other axis in order to create a large number of concentration combinations over the grid. So depending on the bacterial response, whether you see a cloudy or whether you see growth suppression overnight, you will come up with different categorical interpretation, either by looking at the patterns of the zones, or whether if you want to be numerically, you're more into objective assessment, then you can come up with an FIC index, which is really a standard formula looking at how well two agents work together. I think this is pretty well established in the field already. Although that there have been hundreds, if not thousands, of papers published on the FIC index, I still find it not very useful for the following reasons. Number one is the basis for the construct FIC index is really based on a implicit assumptions that the two agents are involved. So in this case, it's the beta-lactam and the beta-lactamase inhibitor. They have similar or even linear concentration effect relationship. Okay, I won't spend too much time on it, but just there is an implicit assumption on the mathematical formulation, how this will work. And the idea was this was initially developed by environmental toxicologists, this FIC index. And essentially, we were looking at very minute quantities of environmental toxin in soil and in environmental samples so that you can pretty much make those assumptions. But when we are looking at this clinical problem with multi-drug resistance and trying to counter this problem with combination therapy, we are not using minute quantity of antimicrobial agents. In fact, I would argue that we were using as much as we can if we can get away with it in terms of safety to address this problem. So many of these implicit assumptions may not hold true under many circumstances. And there is also a computational hurdle in the obvious fact that the beta-lactamase inhibitor does not really have intrinsic activity. Now, I'm going to go back one slide to show you that if you want to compute the FIC index, you need to have MIC for agent A, and you also need to have MIC for agent B. Okay, but if one of the agents, in this case the beta-lactamase inhibitor, is not active, you can't come up with a MIC for the BLI. So this doesn't really work computationally. And my major problem is really doesn't really matter if you can get this to work, but is it predictive for clinical outcomes? And there are thousands of papers published on this, but very few has actually linked the use of this test to clinical outcomes in terms of its predictability. And actually, there are two papers here that actually explicitly show that FIC index did not work. So we need something a little bit better than the conventional standard platform. So this is the way I see this when I first started to tackle this problem. If you're looking at the bacteria, the practice of infectious disease in microbiology is unique in the sense that we have the ability to recover patient-specific samples. We don't treat asthma the same way like a pulmonologist, because we have the ability to obtain a patient-specific sample, and based on what we find, the pneumonia could be treated differently based on susceptibilities and resistance mechanisms. Okay, so we are a lot more personalized in our treatment algorithm. So once we have the pathogens, we have to consider several things. Obviously, that the bacterial density is a key factor. I mean, obviously, there are infections that are associated with a higher bacterial burden. I mean, clinicians know this very well. The way you handle an intra-abdominal infection is very much different from the way you handle a uncomplicated cystitis. Simply, there are different amounts, different number of bacteria that you're going to be encountered in the site of infection, and especially when these bacteria are producing enzymes. So you can see that you can have a very much different hurdle that you need to overcome. And also, once you have figured out that these bacteria are harboring or expressing enzymes, the enzyme types also matter. I mean, simply pick a carbapenem. There are several genetic classes of carbapenems. You can make categorical interpretation that you are more concerned with a metallo-beta-lactamase because they have much more higher efficiency of hydrolyzing carbapenem than perhaps say some of the oxa type, the serine-based carbapenems. So you can make categorical inferences based on what enzymes and enzyme variants that you're able to recover, making an educated guess as to how much of a burden that we need to overcome, and also on the enzyme efficiency. But I also want to point out a fact that for those of you who are practicing in the field, you would often come to realize that the bacteria is not mandated that they can only have one enzyme and one enzyme only. Unlike that you read in the papers, oftentimes in actual practice in the field, bacteria is not surprising to see that a bacteria could harbor multiple enzymes. So they could have a ESBL, a carbapenemase, and a metallose all at the same time. Is not unheard of. So, and even though with that knowledge, we still do not have a good handle on each individual enzyme and the level how much they are being expressed. Most of us detect this enzyme presence by PCR looking for the gene, but we have no idea. We don't have a good, robust way to quickly figure out the level of enzyme expression. And all these factors would come to me as a way to look into an analogy of trying to jumping over a hurdle. So this would come up with have an idea of assessing the target so that we know how high the hurdle is, so that we can know how high that we have to jump. Okay, without a proper assessment, you're simply, the analogy will be, if you want to jump over a hurdle, you need to size it up. You need to know how high the hurdle is so that you can plan your run, how far back you have to run, how fast you have to run, and how high you have to jump in order to clear them. So the analogy is to get a proper assessment on the bacteria so that you can define the burden of the hydrolytic capability that you need to nullify. Now, on the other hand, that you need to consider the drug factor. And these are the standard stuff that people are very familiar with on the potency of the inhibitor, how well can it reverses MIC, and the potency of the drug. Remember, we actually need both agents to work together. So you need to be careful about ensuring that the concentration at the infection site, they probably should have similar binding to serum proteins. And the metabolic route, you don't want to have one drug that is clear from one route and the other one is being metabolized, and you don't end up with both agents at the same time at the site of infection. And for the most part, people have been trying to match these agents based on pharmacokinetics, especially with elimination half-lives, so that you can have a way to ensure that you have a decent shot at ensuring you have adequate concentration, or at least you have some concentration at the target site. Without a proper evaluation like this, then it is not very wise if you're looking at the hurdle. And if you don't do your homework well, you could easily end up with the hurdle failure because you don't know how high the hurdle you have to jump, and if you simply jump, you may not be able to clear the hurdle every single time. So this is in a nutshell where I came from in order to start this area. Now, obviously, for those of you in the audience who are computationally more savvy, you're not afraid of mathematics, there's a better way to do this based on differential equations and co-modeling of multiple behavior. I'm just going to very quickly go through this. You have number of bacteria that you can track, and if you leave them alone because they have good environment, nutrient, and oxygen and space, they would intrinsically grow at a certain rate. And by introducing bacteria that they would have, by introducing antibiotics, you're basically inducing a cure rate to nullify the natural growth rate. So you can set a mass balance equation showing how much antibiotics you need in order to keep the bacteria from growing, or if you have more killing than growing, you can actually suppress the bacteria. But in this scenario, we have the bacteria able to produce an enzyme. Okay, and this is just a simplistic representation of this one enzyme. If you have multiple enzymes being expressed, then you may need multiple circles representing each of the enzymes, like Oxford, ABC, VIM1, and NDM1. So you need multiple enzyme boxes to be able to track the amount of enzyme being produced. Of course, the more bacteria, then you have more enzymes, and these enzymes are designed to nullify beta-lactam so that you would have a negative function on the cure rate of the bacteria. In a negative way, you are allowing the bacteria to proliferate. And now we have added on the third party. The complicated issue is now we add an inhibitor to inhibit the enzymes so that the enzymes cannot inhibit the beta-lactam, and the beta-lactam cannot inhibit the bacterial growth. So this is very complicated. I think many of you can do this in the laboratory and when you have the luxury of time, but I just don't see this is going to be very applicable at the bedside when you are trying to struggle with time and you have a finite period of time that you come up with a solution. I'm just not, I know that people have published on this approach and I have done so as well, but I don't see this as a very practical solution, especially when looking into implementation on what to do next for the patient. All right, so then now we have a struggle of too many problems and we can't solve this. But the way we approach this is, well, depending on enzymes, depending on enzyme type, depending on efficiency, at the end of the day, they do one thing, they break down the drug. So why don't we take a more pragmatic approach in order to try to get a rough estimate of the hydrolytic capability? So this is one set of data that I'm showing you is essentially looking at a substrate, how fast it is being converted to a colored product. So you can see that despite all these bacteria are known to have a ESBL, okay, they clearly do not have the same hydrolytic capability because either they have multiple enzymes or because they have different enzyme level expression, okay, and we don't really know what is which. But when we try to correlate this to the MIC, we thought that there was a striking correlation. And we can see that no matter how many enzymes are being produced or what level being expressed, you can actually track as a surrogate of this type hydrolytic capability using MIC as a surrogate marker of overall activity. And that a light bulb basically went on and said, maybe we have a better handle on this instead of trying to track each individual enzyme, what is being produced and how much is being produced. There is a more pragmatic way of looking at function. And the analogy that I try to explain to my students is that if you go to a cafeteria and if you go to the salad bar to get a salad, at the end of the line, you don't really pay by the number of cherry tomatoes that you get or the pieces of cucumber that you get. At the end of the day, at least in the US, we put the plate on the scale and you pay by the weight. Regardless of how many cherry tomatoes you pay, how many onions you have, there could be a numerous combination. Essentially, you use a surrogate marker as a rough estimate of hydrolytic capability. So this is the foundation of what we're trying to do to approach a very, very complicated problem. Right, so now when you talk about clinically, how do we do this testing? Typically, you have a range of the beta-lactam and to be tested with a concentration of the BLI, beta-lactamase inhibitor. But the question is always come up with what is considered, what concentration that we need to test so that the results are going to be clinically and dynamically relevant. People have come up with a range of responses. You can use the fixed inhibitor concentration at the trough. Use this at the peak concentration of the inhibitor. You can use the average concentration of the inhibitor at steady state, or you can lock in with a fixed ratio of both the BL and the BLI. To me, this is kind of a trick question. No matter what you do, you're not going to get very satisfactory results. And the analogy that I'm going to give you is a story of a several blind man trying to figure out what an elephant really looks like. So I am having trouble advancing to the next slide. Let me see if I can do this. Okay, got it. So essentially, this is a story about several blind men trying to figure out what an elephant looks like. But they only have each one of them have a very microscopic perspective of what is really going on. So at the end of the day, everybody is correct, but they are not comprehensive in understanding the overall picture. They can argue all day, all night, and nobody would agree with one another. So I think we are having a very similar situation here that I think instead of trying to argue that I am right, you're wrong, this is better, I think what we need to do is to take one step back and try to understand the whole picture. We need to define the dose-response relationship in a little bit more comprehensively. And this is what I'm going to show you next. And this is arguably one of the two most important slides in my presentation. So this is the time that you need to pay attention. The way we propose to do this is to set up multiple MIC almost in a way that you would do this in a checkerboard assay. So you would have multiple MICs of the beta-lactam here on the y-axis in the presence of escalating concentration of the inhibitor concentration. So you can set this up by automation or by manually, it doesn't really matter, but you get multiple readout of the MIC in the presence of multiple concentration of the BLI. And you are going to get different responses depending on the nature of the bacteria that you're dealing with. I'm going to first start off with the black line. This is pretty easy. Okay, so this will be the scenario where you have a metallo beta-lactamase, but you are trying to test it against tazobactam. Okay, the inhibitor does not really have a chance of inhibiting an enzyme, and so the MIC would remain very high no matter how much inhibitor used. This is pretty much a negative control. You can see that using high concentration of the inhibitor is not going to make an impact on the MIC reduction. And the good example, the positive control, is the green line that I'm showing here. That you have a very dramatic response in the reduction of MIC at a very, very low concentration, and it stays flat across the concentration range that you would expect to see clinically. So I have also highlighted two triangles here representing the range of the concentration that you are likely to see when the drug is given to a animal, or you can use allometric scaled exposure in humans, or if you actually have human PK data, you can also use that to guide what is considered as clinically relevant range. So you can see that for the green line, you get rapid reduction in MIC, pretty much way lower than the concentration that you will see, that you would expect to see clinically. And the concentration, the MIC would pretty much remain flat across the entire concentration range that you see between the two triangles. This would be a good example. This would be a good inhibitor to have, but this is not likely to be seen. I have worked on this for almost 10 years. It's not like it doesn't happen, but you have to create a very drastic scenario, perhaps using a most advanced generation of a BLI going against an antiquated enzyme. So maybe it's like a SHB12 versus AV-bactam. Maybe you would get that kind of drastic response. For the most part, it doesn't happen. For most more realistic cases that you are testing new drugs for more of the contemporary enzymes, this doesn't happen. There's also an intermediate scenario, which I'm showing in yellow here or orange here. Then you can see that there are alternative mechanisms in place. So this would be an analogy that perhaps one is that you have multiple enzymes involved. The tazobactam used can actually nullify the, perhaps say, the SHB or the TEM26, the older integrated enzymes, but it is being mixed with KPC or NDM1. So then you have multiple resistance mechanisms involved. The tazobactam is only able to do a partial reduction of MIC as shown here. So this is also another possible scenario that when you are encountering multiple alternative resistance mechanisms, this is what you will see. I'm going to show you two more profiles that's much more realistic with my experience that working with inhibitors that are being developed. You can see that for the purple line, that you can see that the reduction in MIC is really occurring in between the two triangles. Okay, you can see that this is where the reduction is, and this is right between, which suggests that depending on how much concentration you have in vivo, you are going to get a variable amount of MIC reduction. Okay. This is more realistic with, consistent with my experience that working with several compounds over the years. And you can also see that scenario with the magenta line. This is the line that has been slightly shifted to the right, that at the maximum concentration, you still don't get much of an MIC reduction, but the reduction would actually come slightly beyond that of the anticipated peak. That would mean that you probably won't have a shot using standard doses. You would have to consider dose escalation in order to get more of those effects that you desire. That is the more significant MIC reduction. But the beauty about this is I have described several different scenarios to you, and they all can be described with a single concentration effect model. So there is hope that we can actually get a better handle on the response more comprehensively by looking at the range of the BLI concentration. All right, so this is the other slide that I wanted to highlight to you. This is the new concept that we have come up with on the instantaneous susceptibility. Essentially, this is a concept which basically would address the MIC is not stable over time. Depending on how much concentration of the inhibitor that you have, the bacteria are in different stages, different states of susceptibility. So you can see that when you have a very high concentration of the inhibitor, okay, this is the green line, this is early. You have a lot of inhibitor, susceptibility is expected to be very high. So the MIC is very low because the enzymes are being inhibited. But as you wash away, as the inhibitor is declining over time, you can see that you don't really have enough inhibitor to suppress the bacteria resistance mechanism. The susceptibility actually would reverse in a inverse relationship. So you can see that the dynamic situation that I have described earlier on, that you don't really have a fixed MIC across the board. And you can use this as a framework to guide you as to what is necessary to hold the bacteria in check. So we have come up with an idea of, what if I use a same backbone of the beta-lactam? Let's take one baby step. Okay, we will use a standard dose of piperacillin, and we are just going to try to increase the dose of tazobactam. Can I see pharmacometrically, okay, this is just a computer simulation, can I build a matrix that I can come up with a gradient of response? And you can see that by increasing the dose of the tazobactam, we would have different anticipated peak concentration, and we come up with a gradient of a new PKPD exposure gradient. Okay, so you can actually repeat this exercise. I'm not showing you here, but you can repeat the same exercise holding the same dose of the inhibitor, and you can start changing the dose of the beta-lactam. You can also get a very similar gradient. Okay, so that gives us the framework, the flexibility of the framework, that you can hold one drug at a fixed level, change the other, and you can see the impact on the surrogate exposure. And of course, you can change both. You have the framework to do so. But that is only the pharmacometric solution. We want to take the next step and see whether this has actually have any biological possibility. So we look at a hyperproducer of ESBL. That is basically, if you do the standard testing, you would consider this as a intermediate resistant strain. And we did the MIC profiling. We did basically the MIC, the piperacillin MIC in a range of tazobactam concentration shown here. So you can see that piperacillin the MIC was not maximized at tazobactam concentration of 4. If you further increase out the tazobactam concentration, you're still able to get more MIC reduction beyond what you can see at 4. So then now we actually did a experiment to compare, what if I simply use the same backbone of piperacillin and use 2 different doses of tazobactam? But I have different bacterial responses. And as I've shown you here, that we get very dramatic response by combining 2 agents, simply by changing the dose of the tazobactam. And we repeated this exercise multiple times, and we were able to show that depending on where you have, there is a threshold when you can actually come up with to see how much exposure is necessary to keep the bacteria in check. So we were pretty surprised, and we were pretty pleasant that at least there is a framework that will allow us to go forward addressing dynamic susceptibility changes over time, and also by giving us a tool to evaluate different combination of doses of the beta-lactam and the beta-lactamase inhibitor. We're not quite done yet. I'm just going to show you different categorical scenario. This is the typical scenario that we have to start off with. The MIC is higher than the peak concentration. Okay, so now you don't really need a PKPD scientist to tell you that this is a bad situation and you don't really have a shot. The drug doesn't really have a shot in controlling the bacteria. What everybody wants is this. It doesn't really happen. Is you want to give a beta-lactamase inhibitor to have a constant suppression of the bacteria all the time. But unless you are dealing with a very drastic beta-lactamase inhibitor and enzyme combo that it may or may not be clinically relevant, this is not likely to happen. This is what I've shown you earlier on. This is more likely to happen. The susceptibility would actually change over time and in inverse proportion to the beta-lactamase inhibitor concentration. And finally, if you push the dose, you can actually modify this in a different shape, and you can easily see this with a large number of possibilities using computer simulations. And then you can actually experimentally select what you want to do to validate this downstream in the hollow fiber, in the mice, or in the human clinical trial. And finally, we have also want to talk a little bit about optimal pairing concentration. I had mentioned earlier on that currently all the products are basically locked in terms of the agents, so you don't have a flexibility to mix and match. But we find that sometimes you may not actually have the right combination for a particular bacteria that you encounter in the clinical setting. So this exercise was looking at using piperacillin as the backbone therapy and using the same approach, evaluating 3 contemporary BLIs to see if we actually would have a better matrix in helping clinicians to match up what is the optimal beta-lactam, beta-lactamase inhibitor combination. So I'm just going to show you some highlights of that paper about guiding what to combine. So clearly we are not done yet. This is only the beginning. I mean, we're getting some little bit promising data to get ahead, addressing some of the limitations, some of the obvious limitations that was hampering our desire to move forward. But there are clearly many questions that have to be answered in the future. We're looking for customized regimen design for combination, so that can we use the bacteria using objective data to customize what is the best way to combine and what doses to combine for a specific patient. We are talking about agent comparing, whether we can free to use different doses combinations, and whether we have to use dosing frequency. Right now, you are giving both agents at the same time because they are both locked in the same product. I can show you on another day on a range of computer simulations that you can actually consider giving the BL, beta-lactam, and the beta-lactam agents at different times. And sometimes they are worse, and sometimes they're actually better. There are different scenarios that you can create, and you can see them through a range of computer simulations. Obviously, the question is, once we have optimally combined these products, can we actually prevent or delay the resistance emergence? And this is a key question that I don't think we are ready to tackle that at this point, but clearly there's something in mind that we want to keep this at the back of our mind. And obviously, we want the results to be able to be applied relatively quickly. I think the checkerboard design can be actually is amendable to automation, and you can make trays, customize trays, so that you can use the standard framework to do MIC in a relatively short period of time, allowing the results to be shared with clinicians so that meaningful decisions can be made by the bedside. And finally, I don't want to belabor this. I mean, the hurdle is really on the regulatory framework. Would the regulatory agencies, would the colleagues in this agency accept this framework? And this is for us to, the burden of proof is really on us to show them that by doing this in a more comprehensive fashion, we are getting more information, and the prediction that we come up with is more robust and more predictive of clinical outcome. So this is really what I want to show here. I have a couple of backup slides, but I think I'm out of time, and I'm going to save that in case you guys have questions. Thank you very much for your attention.
Thank you, Victor, for your lovely presentation. I think, are we opening to questions now? Sorry, Vincent. Victor, are we opening to questions now?
Yes, we are, Shampa. I'll just move the slide along.
Thank you. So I've got a question here for you, Vincent. Someone has thanked you very much for your fascinating presentation, and they'd like to see if you've got any advice on how you would organise the clinical testing of a non-fixed BL-BLI combination.
I actually have to unmute myself, sorry. So let me try to understand the question. Is looking for advice on how do you test this clinically? Can you repeat the question, please?
Yes. So do you have any advice for us on how you would organise the clinical testing of a non-fixed BL-BLI combination?
Well, the testing part is not difficult. The testing part, like I showed you early on, is really you need to come up with customized trays and of the checkerboard design. So you are going to, instead of just doing this with one concentration of the BLI, you are doing multiple MIC with a range of the BLI concentrations that you think are clinically relevant. So this is to help you to get a range of the response within the clinical relevant range. I think with that design, you are going to get better prediction as to whether the standard dosing is going to work. Knowing that we don't have the luxury of using products outside of the fixed combo setting, I think you can take one step back as to make an assessment whether the fixed combo at the fixed dose is going to work. The comparison is not as robust. You don't have all the possibilities, but you are locking down your assessment based on a handful of known fixed combination, and you still can make limited prediction as to which one of this finite combination is going to give me the best predicted exposure.
Sure. So Vincent, do you still think we're quite far away from being able to design a BL-BLI combination that isn't based on a fixed dose? Because I think where we're going with this is maybe it's not a one dose fits all, and ideally, if you could, you would change the dose. But are you suggesting that's just not possible at the moment?
Well, I would like to address this in a more optimistic way. I don't think we're ready to do this tomorrow, that's for sure. But I think we are getting a handle. I think we are coming out of our naivety that, as you have pointed out, I think that many colleagues that I work with, many colleagues that I hang out with, they recognize that the one dose fix all is simply too naive an assumption. Okay, there are multiple enzymes involved. They have different combination of enzymes, and they have different efficiency, and they have different expression level. It's quite impossible that we can come up with a magical dose and a magical dose combination that fix every single scenario. I think some flexibility is going to be welcomed, but I don't think this is going to be done tomorrow. I think we are getting a handle on how at least one possible way going forward. I think I see this as more optimistic that we are addressing the problem head on. We're not afraid of difficult problems. We're not doing the easy thing. We're doing the right thing, and hopefully with more promising results, we can convince colleagues either on the in practice or in the regulatory agencies that this is a better way to look at these kinds of product.
Thank you. So I have another question here. Again, a lovely compliment, a great presentation. The person asking the question says that they see a similar issue and solution to those seen in malaria and HIV. So two issues there are how the in vitro density of the pathogen actually scales to humans, especially when you consider the sites of action. So, you know, firstly, do you have any thoughts of how that scales? And then do you have any thoughts on how to address the variance when your PD variance overwhelms your variability in PK?
Well, I think let me try to address one at a time. I think that the issue on scalability, is it referring to PK scalability?
I think it was talking about PD.
Okay, so in terms of even the site of infection.
So let me read the, so I think there's two different points here. One's about variability. So one's about variability in your PD, and how do you deal with it when you've got more variability in your PD than you have variability in your PK. And I guess this comes from where you see, you know, such a large amount of variability across many strains.
Right. So I think that this is spot on. I mean, if you can consider the variability in PD is far bigger than what you can anticipate for a PK for the most part, that is a correct statement. So it is more important for us to understand if you have to make a difficult choice. If you want to learn more about the PD, it's going to be more informative than knowing about the variability in PK. Okay, that's why I think in infectious diseases and microbiology, we're spending a lot of time trying to understand what bacteria are we dealing with, what are the likely resistance mechanism, what drugs can be used, and what are the response. I think we are spending the right time, relatively speaking, focusing on what we need to address to get a better handle on that. So spending more resources on defining the bacterial response to different drug is a good way to address the problem. Okay,
thank you. So another question is about when you're conducting combination experiments using, so this is when you're doing your experiments, when you're using varied dosing of the inhibitor, what exposures of the beta-lactam backbone do you use? Do you use humanized exposures or actually fixed concentrations?
Right, so it depends on what stage that you are looking at. So initially, we started with the MIC testing. It's a modified MIC testing as shown in the checkerboard. So essentially, you are making estimates of how well the MIC would look like in the presence of different BLI concentration. That is to gather some input data to as input data for the model. Once we have the model predictions, then we took the next step in validating the exposures, and that's when we use the hollow fiber model that we would actually fluctuate the concentration of both the BL and BLI in a more clinically realistic setting. The concentrations do not stay fixed over time, they decline with a half-life that is likely to be encountered in humans. And of course, the doses may or may not be relevant depending on whether this is going to be a proof of concept study. I mean, we have gone to very, very high doses just to show that this is to show as a proof of concept. But I think for clinical application, you might not have the luxury of going beyond a certain threshold because you can't really give that kind of doses to humans.
Thank you, and I think that your response there directly answers a further question that we had, which was whether you had any comments on toxicity of increasing doses of BLIs.
Right. So I think this is a critical question that has to be addressed at an early development phase. But my point is, I'm not sure, and maybe there are colleagues in the audience that will actually know this better. But I don't think the current products that we have on the market, I don't think that the dose of the beta-lactamase inhibitors is used because of toxicity. It was more like I'm just not privy to the reason why a certain dose was picked, but I'm pretty sure it was not due to a toxicity concern that we are not using a higher dose of the BLI. So I think there's room for further improvement and to look into what is the optimal dose of the BLI that has to be used. And I've shown you that several products, they range from an 8:1 ratio to a 1:1 ratio. So clearly people are not sticking with a conventional dosing ratio as the only option possible.
I think sometimes, Vincent, just from experience, sometimes it's complicated by emerging data during a drug development program. I think sometimes you get fixed into something, you know, once you've got phase 2 data, and it's hard to change after that. Another question is about a specific question about whether you have any experience on using higher doses of ampicillin sulbactam, especially against Acinetobacter.
Well, the answer is no. In the US, we don't actually have a lot of problems with Acinetobacter for some strange reason that I don't understand. I understand that Acinetobacter is a major problem for many colleagues in Asia, in Italy, and in Greece. But primarily, this is not our problem, and I don't have a lot of experience to share on Acinetobacter.
Can you comment on whether there is any advantage to making an individual BLI available for use with active beta-lactam agents? So, for example, a separate BLI to use with Astrienam for metalloenzymes.
I think an argument can be made that in the right hands, if you give a reasonably well-trained clinician the tools, you will give them the flexibility of optimizing combinations. But obviously, I understand there are some concerns that I've heard from colleagues from the regulatory agencies is they are more interested in not doing the most difficult cases, but rather making sure that the majority of the patients in the community are not being harmed. So it's a matter of perspective. I think if you give people the ability to titrate the dose, it's more flexible, but it comes with certain risk of if you have clinicians or practitioners who are not as well trained, there is a concern that they may be using, for example, I mean, I'm just making this up. Maybe maybe someone would not know that the BLI is intrinsically inactive, and errors could have been made by simply treating patients with just the BLI without the beta-lactam. I can see that happening.
If you were, so just before I ask this question, and Vincent, it directly follows on from it. Can I just remind people that you can submit questions through the panel as shown on the slide that's up at the moment. So, Vincent, going back to that point, if you were going to try and develop a standalone BLI product, so you know from scratch, what are the challenges for actually developing it?
I think you brought up early on that if your trial is already logged in at the phase 3 stage, there's not much you can do to further optimize. I think that the right way to do this is to understand the limitations of the current approach and try to do something different earlier. And as simple as trying to do a range of the BLI concentration, so that you can get a more comprehensive understanding of the response. Is the BLI pushable? Is there a limit on toxicity as how much you can push? I think those issues are already making added advantage to the development program. And perhaps as you go further down the clinical development, you can actually have several options. So for example, you may have an option of perhaps say for a certain bacteria that you isolated, the standard dose would be this. But if you encounter a hyperproducers or one of those weird ones that you don't have a handle on, you could potentially have a different product. Instead of locking this as a 1:4 ratio, you can have another product specifically for the hyperproducers at a 1:1 ratio. So it builds in flexibility so that you can be more, you're more able to react to different situations without putting all the eggs in the same basket on the one dose fits all scenario.
Thank you. I have another question here, but I'm afraid I'm not sure I'm following it. It's about what about adverse drug reactions related to it? I'm not sure whether the question refers to the combination or the standalone. I think that might be a tough one to answer, Vincent. Does it make sense to you?
Not really. Try again.
So I've got another one here, which is about Astrienam avibactam and Astrienam zidibactam. And the question is just whether you'd like to comment on either of those two combinations.
It's Astrienam and?
Astrienam combined with avibactam or Astrienam combined with zidibactam.
I think this is a very interesting combination in terms of multiple aspects. Okay, I think the rationale, if I understand it correctly, is you are trying to mix and match the BL and BLI in a way that you will cover and broadening the spectrum of coverage. Astrienam is inherently resistant to the metallose, so this is an advantage, a property that you are trying to exploit. But Astrienam is not very good, again, it's not very stable for the ESBL or the KPC. So you're trying to use a DBO inhibitor to nullify all those non-metallo beta-lactamase enzymes. I think this is the rationale of combining Astrienam with avibactam. I actually like it. This combination makes sense in the current scenario because we don't really have anything that is remotely useful for the metallose. So if you are in a country that the metallose are very prevalent, this makes sense from the perspective of bridging what you want to do with the currently available agents. Of course, this could change when one can come up with an inhibitor for the metallose, and obviously this might or might not be a good idea once we have an effective inhibitor for the metallus. But it makes sense at this point.
Sure, and I don't know if you've worked with Zidibactam. I wonder if the question's looking at whether which one would be a better combination with Astreonam. Do we think Avibactam or Zidibactam?
I don't have personal experience with Zidibactam, but I would encourage you that you can take the idea of the same framework. You can actually, I mean, the method, doing the method is not difficult. I mean, this is basically a automated MIC investigation. I mean, many people can do it. The hard part is not really on doing the MIC. But I would encourage you to go back and look into a small collection of contemporary isolates and actually map this out. If you use Astreonam as the backbone, just like the paper that I've shown you, I've used piperacillin as the backbone, and I combine this with 3 BLIs. And you can use the same framework. I mean, I even listed the reference on the slide already. So you can go back to the paper and look it through closely and see if you can, I have no doubt you can actually replicate that kind of experiment so that you can see for yourself what is going to be more useful for your own development program against the isolates that you are interested in. I don't really have a strong invested interest. One drug is better than the other. My bias here is I'm interested as a PK/PD scientist. I'm interested in developing a better tool so that you can see your data more objectively and a little bit more comprehensively. I think this is where I can come in and help, is to give you a better tool. Which drug turns out to be better, it's not really my primary concern.
Thank you. And can you add any comment on site of action and how you account of this?
On the site of action, I think this would be basically a pharmacokinetic issue that you need to address, whether how well the beta-lactam and the beta-lactamase inhibitor is able to get into the site where you want the drugs to work. The example that I gave in the presentation was, if you have one drug that is being eliminated unchanged in the urine, and you have the other agent in the combination that is metabolized by the liver, then you have a very awkward situation because they are going to be handled differently by the body, and there's a likelihood that you don't see both drugs in the same place. So this is something that you need to address, and also whether you need to do the right biodistribution study to make sure that both agents are getting into the ELF. I mean, if you're talking about pneumonia, they are able to get into the epithelial lining fluid adequately, and how much do you actually have a shot? I think those are important studies that you need to do to give you an idea what the potential of your drug BL-BLI combinations really like. I mean, yeah.
Thank you. So I've come to the end of the questions. I think the window's still open if anyone wants to submit any further questions. But if not, I'll hand back over to Victor. Thank you very much for a really excellent presentation, Vincent.
Shampa, thank you very much for moderating today's webinar. And thank you very much, Vincent, for your presentation and for sharing your expertise with our audience. I'd like to take this opportunity to announce our next webinars in June and July. On 16th June, we will have a webinar from GARDP in which we will present our efforts to bring new treatments for drug-resistant infections to all who need them. We will host another webinar discussing why children need to be prioritized in the fight against antibiotic resistance on 25th June. On the 30th of June, Marcus Seitlinger will present on Phase 1 development challenges in the clinical development of antimicrobials. And on 9th July, Anand Kumar and Kamini Walia will present a webinar on the challenges and opportunities for antimicrobial R&D in low and middle income countries. You can already register for these webinars on the Revive website via the link shown below. So that will be all from my side. I would like to thank everybody for joining today and for contributing to the discussion. I really hope you found this webinar interesting and useful, and that you will join us again in future. Please do make sure to spread the word in your networks and to encourage your colleagues to join as well. Thank you and goodbye.